Our topic was enzyme activity and feedback regulation with specific examples featuring Bethany, Betty, Brittany, Brenda, Carissa, and me, Carol.
Hello, my name is Brittany Florian, and I will first be talking about metabolic pathways. Metabolic pathways consist of specific molecules which involve reactants and will end up becoming a certain product. Metabolic pathway, its definition is the totality of a chemical reaction, and it also causes more interactions between molecules. They also consist of catabolic pathways and anabolic pathways. Catabolic pathway is what breaks down certain molecules into simpler compounds. An example of this is cellular respiration because of the breakdown of glucose when oxygen, carbon dioxide, and water is present. Anabolic pathway is energy that is stored in an organic molecule, and an example of this is the combination of different amino acids to form proteins. From the textbook, it talked about uphill and downhill reactions, which symbolizes the catabolic pathways and anabolic pathways, which is why I added a picture on my slide. When it's going downhill, that symbolizes catabolic pathways, the energy that is being released, and the energy that is being stored is when we go uphill. Now I will be talking about energy. The definition of energy is the capacity to cause change. In the textbook, it mentioned 4 different types of energy: kinetic energy, thermal energy, potential energy, and chemical energy. Kinetic energy is the motion of an object. An easy example to understand this is a person walking. Thermal energy is a random motion of atoms and molecules, and those reactions cause heat. An example of this is a cup of hot chocolate. Potential energy is when matter possesses as a result of its location or spiral arrangement. And an example to understand this is the water that's on top of a waterfall. And lastly, chemical energy. They are available in molecules to release a chemical reaction. An example of this is the use of batteries. And I added a picture on my slide to demonstrate the different energies being used. Thank you.
My name is Bethany Future, and I'm going to be describing the first 2 laws of thermodynamics. So the first law, the initial law, is alternatively recognized as the principle of energy conservation. So when a bear decides to eat a fish, all the chemical energy from the fish turns to kinetic energy, which gives the bear energy to move around from place to place. So every time the energy is transferred or transformed, the entropy of the universe rises. So it goes up. For example, when the bear runs, it's not just about the speed. Entropy increases, which rises the heat and helps the bear break down the food, which speeds up the bear's metabolism.
Hi, I'm Krista Merweed, and I will be talking about free energy changes in exergonic and endergonic reactions. An endergonic reaction is basically an artificial chemical reaction that involves the absorption of free energy from the surroundings. This type of reaction essentially stores free energy within molecules, leading to an increase in G, which becomes positive. An exergonic reaction refers to a natural chemical reaction that results in a net release of free energy. This occurs as the chemical mixture loses free energy, leading to a decrease in G, which becomes negative. Free energy is pretty much when a reaction deliberately moves away from equilibrium and the free energy will increase.
Hi guys, my name is Betty Gould, and I'm going to be talking to you about ATP powers cellular work by coupling exergonic reactions to endergonic reactions. So the cell focuses on three main types of work: chemical, which is the pushing of endergonic reactions that would not occur spontaneously; transport, which is the pumping of substances across the membrane against the spontaneous movement; and mechanical, which is the contraction of muscle cells and movement of chromosomes during cellular reproduction. A key way that cells manage their energy to perform the work needed is by energy coupling. ATP is responsible for organizing the majority of coupling in cells and acts as an immediate source of energy to power the cellular work. How does ATP provide the energy that performs the work? When ATP is hydrolyzed in a test tube, it heats the water surrounding it. This was discovered based off a experiment that scientists did in a lab. They also discovered that the same process of heating ATP can be beneficial in an organism. So placing the ATP in a hydrolyzed system in the organism heats it up the same way as it would the water surrounding it, except instead it works in the organism. However, the generation of the heat alone may be dangerous and inefficient. There's not enough heat that's going to be able to work for the organism, so it ends up not being as helpful as it would need to be. Instead, the cell's proteins harness the energy released to perform the three types of cellular work that I previously mentioned: the chemical, transport, and mechanical work. The organism at work uses the ATP continuously, but ATP is a renewable resource that can be regenerated by the addition of phosphate. So as the ATP is getting used, it is also being regenerated as phosphate is being added to the cells. The shuttling of inorganic phosphate and energy is called the ATP cycle. This cycle repeats over and over and over again, which helps the energy needed for the cells to perform this work.
Hello, my name is Brenda Tavares, and I'll be talking about enzyme regulation. An enzyme is a macromolecule found inside of a cell. There are many types of enzymes with different functions. Location inside of a cell will vary, but they are usually found in lysosomes. They will play a part in helping chemical reactions occur. Substrate intake in the active site depends on the regulation. Allosteric regulation consists of activation and inhibition, also known as regulatory molecules, which have a binding effect on enzymes. This regulation moves around protein functions. Activation will bind and support the enzymes, and ADP is an activator for enzymes. Inhibition will make enzymes inactive, so no substrate intake will occur. This can also be thought of as a pedestrian light, where green would be for go as in activation and red would be for stop as in inhibition.
Hey guys, my name is Carol Cisneros and I am going to be talking about cofactors, and they are basically non-protein chemical compound that helps for catalytic activity to allow an enzyme to carry out its function. It is attached to the enzyme tightly bound or loosely bound, and there are two types of cofactors, which are inorganic molecules, also known as minerals, and organic molecules, which are also known as coenzymes. An example of inorganic molecules would be like zinc, iron, and copper in ionic form. An example of organic molecules would be vitamins such as NAD+, which contains niacin, and FAD, which contains riboflavin.
Active site and catalytic cycle of an enzyme. In the majority of enzymatic reactions, the substrate is anchored within the active site through weak interactions like hydrogen and ionic bonds. Within this site, specific amino acid residues facilitate the conversion of substrate to product, after which the product is released. Subsequently, the enzyme becomes available to bind another substrate molecule. A single enzyme molecule often processing approximately 1,000 substrate molecules per second. An enzyme has the ability to transform one or several molecules that react into one or more resulting molecules. In the illustration provided, this enzyme changes two substrate molecules into two product molecules.
Inhibition of enzyme activity. There are three types of binding, which are normal binding, competitive binding, and non-competitive binding. In normal binding, the substrate gets attached to the enzyme. Then the substrate turns into products, which then end up leaving the active site, and then the process ends up repeating itself. In competitive binding, the competitive inhibitor interferes the active site, so the substrates get blocked from binding to the enzyme. In non-competitive binding, the non-competitive inhibitor is attached to the bottom of the enzyme, making the enzyme change shape, so then the substrate is unable to bind onto the enzyme.
On this slide, I'll be talking about feedback inhibition. Feedback inhibition is a method of metabolic control in which the end product of a metabolic pathway acts as an inhibitor of an enzyme within that pathway. Feedback inhibition typically operates via a structure known as the allosteric site on enzyme. This site alters the enzyme's conformation, thereby influencing the activity of the active sites. In feedback inhibition, the attachment of the end product to the allosteric site hinders or halts the enzyme's function, reducing the production of new end products. As the levels of the end product decrease, the enzyme encounters fewer molecules of the end product, leading to a comeback in its activity.
So these were our references that we ended up using for our topic.